Welcome to the deep dive. So picture this. For less than $100, right now, you can get the entire sequence of a human genome, every single letter.
Yeah, 3 billion letters. It's kind of mind-blowing when you think about it.
It really is, almost magical. But here's the catch. Even with all that data, most of those billions of letters just sit there, like these inscrutable strings of A's, C's, G's, and T's on a server somewhere.
Exactly. And biologists, well, they know the answers are in there. Answers to rare diseases, uh, how to make crops more resilient, even how our brains develop.
But the code itself, it keeps its secrets pretty well hidden. It's not giving up the meaning easily.
No, it really doesn't.
Yeah.
And it's way harder than reading computer code, for example. Someone used a great analogy. Think of DNA like an operating system, okay? But it's an OS that rewrites itself every single time a cell starts up,
Wow, okay, so constantly changing.
constantly. And genes, the parts that actually do something, they might be hundreds of thousands of bases away from the little switches that turn them on or off.
Right, these distant regulators.
Exactly. Or, think about this. One cell might completely silence a gene, just turn it off, while the cell right next door desperately needs it.
And tiny changes can have huge effect.
Oh, massive. A single extra A, just one base, can make a whole chunk of a gene, an exon, just disappear. Poof, gone from the final message.
That level of complexity, you
Yep.
can see why traditional analysis struggled.
Absolutely.
Yeah.
And the earlier AI tools, I mean, they were a step forward, but they had their own issues.
Right, they were very siloed, weren't they? Like different tools for different jobs.
Totally. You had Deep C, maybe looking at chromatin, Splice AI trying to predict splicing, Orca mapping 3D loops.
So researchers ended up having to kind of patchwork everything together.
Pretty much. Stitching together predictions from 5, maybe 10 different models. And you're still left wondering, okay, which one's right? How do these things even interact? It was fragmented.
Yeah, a piecemeal approach to what's really a single interconnected system.
Exactly. So what the field desperately needed was like a unifying lens. Something that could see the local details, the long-range interactions, how all these different signals connect.
All at once.
All at once. And crucially, without needing, weeks of supercomputer time just to get one answer.
Okay, so that sets the stage perfectly for AlphaGenome. This is positioned as that unifying lens, right?
That's the idea.
Yeah.
It's designed to tackle a whole megabase of DNA that's 1 million base pairs in one go.
1 million, okay.
Yeah. And it sees every single base, they call it single-nucleotide resolution. And from that 1 megabase view, it predicts over 5,000 different things.
5,000.
Across 11 different types of genomic data or modalities,
Yeah.
all in a single shot.
That doesn't sound like just tweaking the old methods. That sounds like a leap.
It really is. It moves AI in genomics from being maybe a hopeful draft to something genuinely practical, a comprehensive instrument you can actually use.
So, our mission today, for you listening, is to really unpack AlphaGenome. We want to get into what it is, how they built it, this engineering leap.
Right, and where it really shines, but also, where it still has some rough edges, its limitations.
And what does this unlock? For labs, for hospitals, for biotech startups, basically for anyone asking, what's the state of AI in genomics right now?
Exactly. This deep dive should give you a real shortcut to understanding this development. It's pretty groundbreaking.
All right, let's get into it. How did they actually build this thing? What's the engineering breakthrough behind AlphaGenome, especially this idea of getting long views without fuzzy edges?
Okay, so historically you had these two main paths, right?
Yeah.
The older convolutional models, CNNs, they were okay for shorter sequences.
Like how short?
Maybe 10,000 bases, 10 kilobases,
Right.
chops, because their receptive field, how far they could see, just didn't expand fast enough for really long DNA stretches.
Okay, limited view. What was the other path?
Transformers. Now, transformers are great at grasping context over long distances in theory,
But there's always a but.
but they are memory hogs. They just gobble up computational resources.
Mhm.
So older systems using transformers had to kind of cheat. They'd chop the output into these really small 128 base chunks.
So you might see the forest, but all the trees are blurry.
Precisely. You
Yeah.
lost the fine detail. So AlphaGenome needed to crack both problems, get that long-range view, and keep the single base resolution.
So how does their hybrid approach work? It sounds like they combined ideas.
They did, quite elegantly.
Wow.
It starts with what they call a sequence encoder. This uses 7 stages of convolutional layers that sort of downsample the sequence.
Okay, what does that achieve?
Well, first, these layers are good at spotting small, important patterns, like motifs in the DNA. And as they do that, they shrink the sequence length. making it manageable.
While adding more information layers, the channels?
Exactly. You make it shorter but richer.
Yeah.
That gets you partway towards the long view.
But the real long-distance communication comes next.
Right. That happens in the transformer tower. This is where the clever multi-head attention layers come in. Think of it like a conference call for different parts of the DNA.
So elements far apart can talk to each other.
Yes. An enhancer sequence, maybe hundreds of thousands of bases away, can directly interact, computationally speaking, with the gene's promoter, its start switch.
And it keeps track of which part is which.
Critically, yes. They don't lose their token identity.
Yeah.
Each piece maintains its meaning even when interacting across vast distances.
That's impressive. But DNA isn't just a flat line, right? It folds up in 3D. How does the model handle that?
Good point. That's where the pairwise interaction blocks come in. These are specifically designed to figure out which parts of the DNA are likely to be physically close to each other when it's all folded up inside the nucleus.
So they build those 2D contact maps we see.
Exactly. They predict the 3D structure, essentially.
Yeah.
And then, to get back to that sharp focus, the no fuzzy edges part, there's a UNet-style decoder. This uses skip connections, basically paths, for information to bypass some layers to bring back all the fine-grained base pair detail from the initial encoder stages.
So it puts the high-res detail back onto the long-range picture.
You got it. That's how the final prediction heads can operate at that single base resolution.
Okay, that architecture makes sense. But processing 1 million bases at that detail, surely the training cost is enormous. Sounds like it would break the bank.
You'd think so, but they have a clever trick for efficiency during training. They use something called sequence parallelism across multiple TPU chips, 8 TPU v3s in this case.
Splitting the sequence across chips.
Yeah. It keeps the memory requirements for each chip manageable, even while the whole system is looking at that entire megabase window. It's a smart way to handle the scale without needing one giant, impossibly expensive chip.
Very clever. Now, I also heard about a 2-stage learning approach. That sounds key for making it practical. Can you walk us through that?
Sure. It's a really neat strategy for balancing accuracy and efficiency. Stage 1 is pre-training.
Okay.
Here, they train multiple models, they call them teachers, using a technique called four-fold cross-validation. And they train them on absolutely massive public data sets.
Like which ones?
We're talking ENCODE, GTEx, the 48 Nucleon Project, Phantom 5. Huge collections of real-world experimental data about gene expression, how DNA is packaged, where proteins bind, 3D structure, all sorts of stuff across many cell types.
So these teachers become experts by learning from this wealth of public knowledge.
Exactly. They build a really robust general understanding of how genomes work. Then comes stage 2, distillation.
Distillation. Okay, what happens there?
They take 64 of these pre-trained expert teacher models, freeze them so they don't change anymore, and then train a single student model.
And the student learns from all the teachers.
It learns to predict the average output of all 64 teachers. It's like getting the consensus wisdom of the whole expert panel distilled into one much more efficient model.
And what's the practical payoff of that student model?
It's incredibly fast. This student model can take a genetic variant, run the analysis, and predict its likely impact in less than a second.
Less than a second. Wow.
Yeah, and this whole 2-stage process, it massively cuts down the computational cost. They use about half the computing budget needed for Informer, which is a previous top model.
And the accuracy, did they sacrifice accuracy for speed?
Nope. That's the amazing part. It still beats Informer on accuracy benchmarks. So it's cheaper, faster, and more accurate. But that's a huge win for making this kind of powerful AI accessible.
That really is remarkable. Cheaper, faster, better is the trifecta. And the sheer range of things the single model predicts. The modalities at a glance. It's kind of staggering.
It really is. We're talking 5,930 different prediction tracks for humans, or over 1,100 for mouse model.
And what kind of tracks are these?
Oh, it's a huge list. You've got RNA expression levels, how active a gene is, things like CAGE and ProCap, which measure transcription start sites very precisely. Then all the splicing information. Where the splice sites are, how often they're used, which junctions are created. Crucial stuff.
Right, the cutting and pasting of the RNA message.
Exactly. Then you have measures of chromatin accessibility DNAs and ATAC6 signals, showing where the DNA is open for business. Histone marks those chemical tags on the proteins that package DNA. Transcription factor binding sites where the key regulatory proteins actually latch on.
So open windows, and who's climbing through them?
Good way to put it. And even the high-C contact maps showing the 3D looping and interactions across the genome.
All from one model.
All from one model, one API call. Seriously, no other system offers that kind of breadth in a single package. That's its unique selling point and honestly a massive relief for researchers. No more juggling dozens of different tools and outputs.
That simplification alone is huge. Okay, so let's talk results. The claim is benchmark results that shift the goalposts. Is that just hype, or do the numbers back it up?
Oh, the numbers are pretty compelling. Across the board, AlphaGenome shows really strong performance. It won 22 out of 24 texts focused on predicting those genome tracks we just talked about.
Okay, nearly perfect there.
And maybe even more impressively, on tests predicting the effect of genetic variants, it won 24 out of 26 comparisons.
Wow. So it's good at predicting the baseline state and how changes affect it.
Exactly. And here's the kicker. It often beats specialized models, tools that were designed to do only one of those things really well. It beats them on their own turf.
So the old idea of a multimodal model being a jack of all trades, master of none.
That's out the window. This suggests multimodal learning can actually lead to being a master of many, perhaps because the model learns synergistic relationships between different data types. It definitely shifts the way we should think about building these AI models for genomics.
Okay, that's a fundamental shift. Can we get more specific? Give us some concrete examples of where it outperforms other tools, numbers that really make a difference.
Sure, let's take gene expression levels. Compared to Borzoi, another strong model, AlphaGenome showed a 17.4% relative improvement in how well its predictions match real experimental data that's measured by Pearson correlation, where higher is better, 0.54 versus 0.46.
Okay, a solid jump in predicting how much a gene is on.
Right. Or look at splicing, predicting splice donor usage, which is critical. Compared to Pangolin, it got a 12.7% relative gain in LPRC, that's area under the precision recall curve. Again, higher is better, 0.80 versus 0.71. Better at finding those key splice signals.
That's important for disease variants.
Definitely. What else? Predicting the direction of change for ATAC-seq-QTLs, basically, does a variant make chromatin more open or less open? It showed an 8% relative gain in accuracy over ChrompNet, hitting 0.74 accuracy versus 0.68.
So more confident predictions about accessibility changes.
Yep. And even for those complex 3D high-C contact maps predicting genome folding, it got a 6.3% relative gain over ORCA and Pearson correlation, 0.55 versus 0.52, and maybe just as important,
It runs faster.
4 times faster when just making predictions in inference mode. That speed makes a huge difference in practical use.
Okay, speed and accuracy gains across the board, even against specialized tools. Let's dive deeper into some of those core modalities. What's the real-world impact, starting with splicing, that molecular editor?
Splicing is so critical.
Yeah.
You mentioned rare diseases, something like spinal muscular atrophy, terrible disease, often comes down to errors in splicing signals.
Right.
AlphaGenome's power here is that it looks at splice sites, how often they're used, the actual junctions formed. It sees the whole splicing picture together.
Integrated view.
Exactly. And in tests against benchmarks like MFASS, which uses known disease-causing variants and ClinVar variants, it consistently performed better than established tools like Pangolin and Splice AI, sometimes by up to 5 F1 points, which is a significant metric for accuracy in these tasks.
And what's that mean for, say, a clinical genetics lab?
It means potentially much faster, more reliable triage for those tricky variants of uncertain significance, or VUS. Thousands of these pop up in sequencing, and figuring out which ones matter is a huge bottleneck. Better splicing predictions can help sort the likely culprits from the harmless ones much faster.
Faster answers for patients. That's huge. Okay, what about gene expression? You said it's subtle but crucial for common diseases.
Yeah, most common diseases like heart disease or diabetes aren't usually caused by one big broken gene. It's often subtle changes in the amount of expression from many genes.
Polygenic effects.
Right. And AlphaGenome is particularly good at predicting EQTL direction. That means, for a given genetic variant, is it likely to turn gene expression up or down?
It gives you the directionality.
Exactly. It showed a 25% relative improvement over Borzoi in getting that direction right. So instead of just saying this variant might affect expression, researchers get a much more confident prediction. This variant likely increases expression of gene X in liver tissue, for example.
That tissue-specific context is key too.
Absolutely. That level of precision helps researchers pinpoint relevant pathways for drug development or understanding disease risk much more effectively.
Makes sense. And then chromatin accessibility and transcription factor, binding the open windows, and the protein stepping through.
What's powerful here is the simultaneous modeling. It looks at the DNase and ATAC signals where the chromatin is open, and the TFTIP tracks where specific regulatory proteins are binding at the same time in the same genomic window.
So it connects the accessibility to the specific regulators involved.
Precisely. It sees the open landscape and who's active in it. This integrated view led to an 8-point accuracy gain on predicting causality BQTLs compared to ChrompNet. That gives a much clearer picture of the actual regulatory mechanisms at play.
And finally, those 3D genome contacts, the high C maps, that used to be a whole separate challenge.
Yeah. Predicting how the genome folds was tough and often required dedicated models like ORCA. But what's really neat is that predicting these high C contact matrices seems to emerge quite naturally from AlphaGenome's architecture, specifically those pairwise interaction blocks we talked about.
So it's not bolted on, it's an inherent output.
Seems like it. And it performs just as well as ORCA, the specialist tool, but again,
4 times faster.
4 times faster in inference. It really pulls these complex, previously separate analyses into one unified, efficient framework.
Okay, the capabilities are impressive. Let's translate this into the real-world playbook. Where can people actually use AlphaGenome right now? What problems does it solve today?
All right, let's tick through some key areas. First, clinical genetics. We touched on this, but the big win is tackling that mountain of non-coding VUS.
Variants of uncertain significance, the bane of many genetic reports.
Exactly. Thousands of them can clutter a report, delaying diagnosis. AlphaGenome lets you screen each one very quickly via a single API call, checking its potential impact across all 11 mechanisms, splicing, expression, binding, etc.
So you can rapidly flag the ones that look most suspicious across multiple lines of evidence.
Precisely. Flag the high-impact hits in seconds, not weeks. Faster, more confident answers for clinicians and patients.
Okay, huge potential there. What about synthetic biology, designing new biological parts?
It really accelerates the design-build-test cycle. Say you want to design a promoter, a genetic on-switch that only works in specific tissue. You can use AlphaGenome in silico, cyclin to evolve and test sequences digitally.
Guided by its predictions for expression, accessibility, splicing.
Exactly. You can iterate much faster digitally before you even synthesize any DNA in the lab. Saves time and resources.
Makes sense. How about agriculture and crop improvement? Field trials take forever.
Yeah, waiting a whole growing season is slow. AlphaGenome can help pre-screen genetic variants in silico. Geneticists can score alleles for things like drought tolerance or disease resistance before planting seeds.
So you prioritize the most promising candidates for those lengthy field trials.
Right. Dramatically shortens the development pipeline for better crops.
Okay. And drug discovery. Linking genetic variants, like SNPs, to the genes they regulate is often tricky.
It really is, especially for non-coding SNPs acting through enhancers. AlphaGenome helps by predicting those enhancer-promoter contacts directly from the sequence, alongside expression effects.
So in the same analysis, you get clues about which gene a distant variant might be controlling.
Yes, that variant-to-gene linking becomes much more robust and efficient, helping pinpoint potential drug targets or understand disease mechanisms better.
This also sounds like it could help other AI genomics companies, maybe smaller startups.
Absolutely. By making the trained student models available, possibly through an API, it lowers the barrier to entry. A startup doesn't necessarily need to build and train such a massive model from scratch or invest in huge GPU clusters.
They can leverage AlphaGenome's power to build their own specialized applications on top.
Exactly. And ultimately, all these applications circle back to that core advantage.
Right.
One model, one megabase context, all those modalities predicted at once. No complex pipeline integration needed.
One-stop shopping for functional genomic prediction.
Pretty much. It simplifies and accelerates the whole process.
So AlphaGenome isn't just an isolated tool. It sounds like it's designed to fit into a bigger picture, an emerging AI ecosystem for genetics.
That's a great way to put it. If you look at the landscape, especially from Google DeepMind, you see these pieces coming together. You have Deep Variant for calling variants accurately from raw sequence data.
The first step.
Right. Then AlphaFold predicting protein structures, AlphaMasense assessing the impact of variants within protein coding regions, Informer predicting expression.
And now AlphaGenome tackles the regulatory regions, the non-coding space, connecting variants to function end-to-end.
Exactly. It fills a crucial gap. So you can imagine a workflow. Sequencers generate data. Deep Variant calls the variants. AlphaGenome ranks them by likely functional impact across all these mechanisms.
And then clinicians or researchers get a much shorter pre-analyzed list to investigate further.
Right. It streamlines the whole pipeline from raw sequence to actionable insight. We're moving from isolated tools to a more integrated AI genomics stack.
That's a powerful vision. But okay, let's be balanced. No model is perfect. What are the current limitations of AlphaGenome? Where does it still struggle?
Good question. There are definitely still boundaries. 4 main ones stand out right now. First, ultra-distal regulation.
Things beyond that 1 megabase window.
Exactly. While a megabase is huge, we know some regulatory elements can act over even larger distances. AlphaGenome can't see those yet. It's blind beyond its window.
Okay, limitation 1, what's next?
Second, cell state nuance. The model learns from the data it's trained on. If a specific cell state is very rare or only exists transiently, it might not be well represented in the training data.
So predictions might be less accurate for those unusual states?
Potentially, yeah.
Yeah.
The model might get tricked. It needs to see examples to learn effectively. Third, personal genome risk assessment.
How so?
While the student model is great at scoring individual variants, it doesn't currently combine the effects of many variants to calculate a polygenic risk score for common diseases. That's a different, more complex calculation.
Got it. So good for single variant effects, not yet for cumulative risk for many variants. And the fourth limitation.
Fourth is complex traits. Real-world phenotypes, how we actually look or function, involve more than just DNA sequence. There's the environment, developmental factors, downstream biology.
Things beyond the direct sequence-to-molecular function link that AlphaGenome models.
Right. It predicts the immediate functional consequences at the molecular level, but not necessarily the final organism-level trait, especially for complex ones influenced by many factors.
But these limitations aren't necessarily dead ends, right?
No, not at all. The fact that there's an open API encourages the research community to fine-tune the model for specific tasks or cell types. And future versions are expected to incorporate things like single-cell data and epigenetic marks like methylation, which should address some of these current gaps.
Okay, so it's a powerful tool with known boundaries, but also room to grow. Let's try to sum up the key takeaways for everyone listening.
Sure. The big things are: 1, it pushes the context out to a full megabase, 1,000,000 base pairs, while keeping that super sharp single base resolution. No more blurry long views.
Long view, sharp focus. Got it.
2, it covers 11 different types of genomic assays, all these different modalities, within that single model. It breaks down those old silos between specialist tools.
One model, many insights.
3, the performance is genuinely state-of-the-art. Those benchmark results, winning 46 out of 50 tests, show it's not just broad, it's deep and accurate.
Top performance.
And 4, the potential for easy deployment, perhaps via an open API and efficient student models, makes this powerful AI much more accessible for both research labs and industry.
Easier to actually use. Okay, those are great takeaways. Now, looking forward, what's the bigger vision here? Where could this lead?
Well, think about combining this with other tech advancements, like those portable long-read nanopore sequencers. Imagine them being used in remote clinics, maybe generating whole chromosome sequences almost in real time.
Okay.
If you pipe that live data stream directly into AlphaGenome, you could potentially get triage results for critical genetic conditions while the sample is still being processed.
Real-time genomic diagnosis, that's incredible.
Well, think about field biologists. Studying endangered species, maybe, they could potentially spot harmful genetic variants right there on site using a portable sequencer, and this kind of analysis could really help conservation.
Wow, and for crop breeding.
Maybe designing custom promoter libraries, those genetic switches tailored for specific seasons, or changing environmental conditions, iterating between planting cycles. Much faster adaptation.
And you mentioned generative AI models, earlier ones that might design DNA sequences. How does AlphaGenome fit there?
It could be the crucial critic or editor. As AI starts generating novel DNA sequences for synthetic biology, AlphaGenome could evaluate them in silico, ensuring they're likely to be functional in a real biological context, not just theoretical code.
Keeping the generated sequences grounded in reality.
Exactly. What's really profound, I think, is how models like this that finally bridge the gap between long-range context and fine-grained detail are changing the whole equation.
How so?
The social contract between sequencing and understanding is shifting. We generate sequence data so fast now. For years, the analysis and interpretation lagged way behind.
The data piled up faster than we could make sense of it.
Right. But now, the potential for understanding, for annotation, is starting to catch up to the speed of sequencing itself. The answer to what does this sequence mean can potentially come almost as quickly as the sequence reads themselves.
That's a fundamental change. So the genome used to be like this book of riddles, right? Billions of letters, but the meaning was hidden, like invisible ink, sentences scattered everywhere.
Yeah, that's a good analogy.
An AlphaGenome is like turning on a really bright lamp. Suddenly you can see the letters clearly, but more importantly, you start seeing the grammar, the connections, how sentences across different pages link together.
Exactly. It brings the structure and the immediate meaning into focus. Now, there are still footnotes, right, about the environment, development, things outside the raw sequence.
There's complex traits and external factors.
Right. Those are still being figured out. But the main sentences of the genome's language, they're becoming much, much clearer.
And that clarity, that's the spark, isn't it? For researchers, for doctors, for innovators, and, for you listening, for anyone curious about this stuff, that clarity is what's going to ignite the next wave of genomic discovery.
Absolutely. The potential is enormous.
So the final thought for you to ponder. With this new lamp illuminating the genome's text, what new riddles, what previously unanswerable questions are you most excited to see solved next?